Hello, everyone. Welcome to today's webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi, and I'll be hosting today's session on the value of surveillance data in defining the medical need for new antimicrobials. Revive is GARDP's education and outreach program. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All of our webinars are recorded and freely available to view after the live session on our website, revive.gardp.org/webinars. You can also read our series of articles known as Antimicrobial Viewpoints, where experts discuss various topics within the field. We also have a resources section that includes the Antimicrobial Encyclopedia. Here you can read important terms, and some of these also include videos where experts give further explanations. As always, today's presentations will be followed by a Q&A session. You can submit your questions at any time during the webinar as shown on the slide. We will address the questions after presentations and do our best to respond to as many as possible. If your question is for a specific speaker, please do indicate your name at the beginning of your presentation, of your question. This webinar was developed in collaboration with Vivli. Today's speakers are Morrissey, Rebecca Lee, and Patricia Bradford. Our moderator today is Fernanda Lessa. Fernanda Lessa is the Chief of the International Infection Control Branch at the United States Centers for Disease Control and Prevention. She is an infectious disease trained physician with a master's in public health from the University of California at Berkeley. She has extensive experience in healthcare epidemiology and has led population-based surveillance programs in the United States, as well as in Latin America, Africa, and Southeast Asia. She has also overseen the implementation of the Global Action in Healthcare Network for rapid detection, prevention, and responses to antimicrobial resistant threats in healthcare settings globally. Welcome, Fernanda. I now hand over to you to introduce our first speaker.
Thank you so much, Victor, and good morning, good afternoon, good evening, depending on where you are. It's my pleasure to introduce Dr. Ian Morrissey. Dr. Morrissey is the founder of Antimicrobial Focus. He was Chief Scientific and Development Officer at IHMA Europe, SAL, which supports all phases of antimicrobial drug development through dedicated laboratories and expert scientific personnel. Dr. Morrissey received his PhD from the University of London and his MBA from the Imperial College London. He's a voting member of the Clinical Laboratory Standards Institute, CLSI, Veterinary Antimicrobial Susceptibility Standards Subcommittee. Dr. Morrissey is also involved in the British Society for Antimicrobial Chemotherapy Standing Committee on Antibacterial Drug Discovery and Development, and is a fellow of the Royal Society of Medicine. Over to you, Dr. Morrissey, for your presentation.
Good evening, and thank you for joining this webinar on susceptibility surveillance studies and antimicrobial development. What do we mean by surveillance? Well, here's a definition here from the Collins English Dictionary, and basically it means close observation, which in this case is close observation of antimicrobial resistance. Well, why do we perform antimicrobial resistance surveillance? Well, you can see here from this slide that ever since the beginning of antimicrobial chemotherapy in the 1940s with sulfamides and penicillins, that you can see very soon after these antibiotics were introduced to the market, the first reporting of resistance was made. And you can see this has continued over time with all classes and types of antimicrobial agents. So we perform antimicrobial resistance surveillance because we know at some point or another resistance to a new agent will develop and resistance to existing antimicrobial agents will evolve over time. So what actually makes resistance occur in the first place, or drives resistance in the first place? A lot of it is down to direct human activity, whether it's the inappropriate use or dosing of antimicrobials, whether it's poor hygiene or insufficient infection control. In addition, travel can cause antimicrobial resistance to spread, and there's a lot of documented publications there of people going on holiday to certain regions of the world and bringing back metallo-beta-lactamases, for example, or the movement of penicillin-resistant pneumococci. So all of these human activities can help push forward antimicrobial resistance. But also there is a biological element as well. Evolution itself, as we know from Darwin, survival of the fittest, spontaneous resistance can occur due to appropriate mutations within the bacterial cells, and this allows the bacteria to grow if they have this advantage, this selective advantage, in the presence of antimicrobial agents. Also, if there are existing resistant organisms already in the environment or resistance to closely related antimicrobials, then this will also assist in driving resistance further. And also there is often a link between resistance and virulence itself. So often those organisms that have the capabilities of causing serious infections have virulence genes on motile plasmids that also can pick up resistance genes. So when one is doing surveillance, you have to be very cautious, very aware of where you're actually performing your surveillance. To think of a very common infection, urinary tract infections, there's a lot of difference in the type of organism causing a simple, uncomplicated UTI like cystitis compared to something like pyelonephritis, which would be a complicated UTI. So one would expect that the E. coli organisms successful at causing complicated UTIs could well be very different to those that are causing simple cystitis. And therefore, if there is a link between resistance mechanisms and the virulence that allows these organisms to cause different infections, then one would expect that resistance itself could well be different between these types of infection. So these variables that we tend to look at are based on geography. So we find that resistance varies from region to region, from country to country. The pathogen itself, and also the site of infection itself. So when we perform surveillance studies, these are the three main factors that we are looking at. Where is it? What pathogen is it? What's the infection site? There are different types of resistance surveillance data, which can be split into generally three types. One is hospital surveillance data, which is vitally important and is a collection of data from individual institutions or maybe types of hospitals within the same hospital group. And these are local antibiograms where there is a reporting of susceptibility of key pathogens with key infections, and also broken down by ward to see what the susceptibility is like. And often this type of reporting is mandatory to certain health authorities. But this is vital important information for the hospital because it helps them guide the empirical therapy that is more often than not used when treating an infection. So if you know, for example, that your intensive care unit has a particular issue with a multi-drug resistant Klebsiella pneumoniae, for example, or Acinetobacter baumannii, you can guide your therapy on the basis what you predict might be causing this new infection. It's incredibly important, but not really so important for companies developing new antimicrobial agents. Although one might want to work with a local hospital or a friendly KOL, that's a different thing. But the type of data at a very local level is not so useful for a drug developer. What is more useful is the other two types of surveillance data. And one is what I call generally government agency data. And by this I mean the various surveillance activities performed by the World Health Organization, such as GLASS and CSER, and also the European surveillance network, the EASnet. These data are very useful to give people an idea of particularly the geography, how that may affect the level of resistance. And then there are other types of surveillance data which are performed by companies themselves that are developing new antimicrobial agents. And I'll talk a little bit more about this in a moment. But basically, these are data that have been generated for a particular purpose, mainly for the registration of a new drug. But the data of all of these surveillance studies also contain interesting comparators. So it gives people wanting to plan future studies useful data to at least look at and try and decide how they will design their own surveillance study. And some of these data are available online. And the Vivli surveillance activities, which is a collection of virtually all pharmaceutical company surveillance data sets, is a very good resource. I won't say any more about that because my other two co-presenters will be going into this in a lot more detail. But other data that are available online are data from JMI, the Sentry surveillance study, and also the Pfizer Atlas database, which is MIC data that was originally generated by IHMA in the US and in Europe. Both these websites are quite similar and they require registration, but beyond that, one is free to have a look at the data and see what interesting susceptibility profiles can be found. Then obviously, as I talked about a little bit a few seconds ago, a new sponsor themselves will ultimately have to perform their own surveillance at some point or another. Here's an example of hospital antibiograms. And this is just a single example I chose because the data has been published, and it's from a very famous children's hospital in London, Great Ormond Street Hospital. And the sort of data that hospitals collect, in this particular case, it's looking at methicillin resistant and methicillin susceptible Staph aureus. And you can see here in this graph, here's the prevalence of Staph aureus in the top line. You can see it wobbles about a bit, but is kind of consistent over time, over a number of decades. But you can see here, if you look at the split between MRSA and MSSA, up until around about 2014, 2015 or so, the prevalence of MRSA was pretty constant. And then there was a sudden increase in this prevalence of MRSA. So what they did in this particular study, and also is relatively common in hospital antibiograms, is to not just look at susceptibility data, but also look at molecular data. In this case, is looking at the clonal type or the clonal complex of Staph aureus. And you can see here that something significantly changed, where there became a more diverse presentation of clonal complexes. And within this diverse collection, maybe these were more dominant, more virulent strains that were able to become more prevalent over time. So these types of data are very useful, as I said earlier, for empirical therapy, but also for infection control and that type of activity. As far as government agencies are concerned, and here's an example from the EU EASnet program. These data are published on a regular basis, and part of these data are very nice heat maps that indicate the prevalence of susceptibility within Europe. And this example here is looking at carbapenem resistance in Acinetobacter. And basically what this is telling us is that those countries that are green are good. Those countries that are getting darker and darker red have higher levels of resistance to these agents. So this is very useful data to give an overall picture, but there isn't the capability of actually being able to dive into the data and analyze the data yourself. And this is where online resources come in. And as I said, we will be talking a little bit more about Vivli later on. Here's just an example here. Here's the GAMI Sentry database. And basically, you can slice and dice the data. You can choose whatever organisms you want to use, whatever genus you want to look at, the geography, and other sort of demographic filters here. I won't read them all out. I'll just sort of flick through. There's a lot of things that you can choose. You can also look at MICs, organisms with particular MICs or resistance profiles. And also you can look to see organisms that have particular molecular profiles, particularly beta-lactamase profiles. Also, you can choose just to say, hey, just give me all the data you got, or you can be selective for particular classes of antibiotic if that's what you're particularly interested in. Once you've selected what you want to see, then you will be given a nice table here of the summary data, and this is just a sort of small example that I chose just to give you an idea. And you can break this down whichever way you want to really be able to compare and contrast susceptibility to specific antimicrobial agents with certain organisms. So it's a very nice resource. As it is the Pfizer Atlas database, similar in the way you select, although with the Atlas database you can have heat maps, which is kind of like the EARSnet that I was showing you earlier, but also the actual data tables themselves. And like with the JMI Sentry database, you can select specific information that you might be interested in, and you also get a nice table showing you the susceptibility data, and also you can get MIC summary data as well. And these are very useful data for your own purposes. So it helps you design your new studies. So here's a flow diagram here. I'm sure many of you have seen very similar type diagrams. Actually, this one I got from the internet, and actually wasn't really referring to antimicrobials, but the process that you kind of go through, at least in a broad sense, is very similar. So government surveillance, so these are like the EARSnet, like the CSER, like the WHO surveillance, are very useful for your concept, your original idea. What's the unmet medical need? What are these published government data telling us is really something that is of concern to them? So things like the WHO critical pathogens, resistance, that kind of thing is a good place to look at to say, okay, there's an opening here. This is something that really is an unmet medical need. But also later on in development, actually in clinical development, these type of heat map data are also useful when you're looking at clinical studies. In particular, if your new agent is focusing on a resistant subset, so maybe something like carbapenem resistant Enterobacterales, you want to know where you want to perform your clinical studies. You don't want to go somewhere that's green on the heat map, because it's going to take you a very, very long time to recruit patients that have CIDs. You want to be going somewhere like Eastern Europe, like Southern Europe, if you're looking at Europe, to find countries, regions that have higher levels of carbapenem resistance that will enable you to recruit your patients and complete your clinical study sooner rather than later. Well, saying that, pharmaceutical surveillance is probably more useful if you can get to the data that's of interest to you available online. For the same reason as the government surveillance, it can really help with clinical development down to the level of what sites you might be able to go to. But also this surveillance data is very useful over the discovery and earlier development, the sort of in vitro studies that might be performed. Because the pharmaceutical surveillance studies, unlike the government surveillance studies, generally tend to keep their isolates. And many of these surveillance studies have these isolates available where you can put well-characterized isolates together into bespoke panels of organisms. So for example, if you're interested in isolates that have metallo-beta-lactamases, it would take you a long time to collect all the weird and wonderful derivatives yourself. But if you go and speak to those laboratories that are performing global surveillance studies, they should be able to put together a panel of interesting organisms for you that you can use earlier in your research than perhaps you would normally do. So it really helps to try and find the lead compound and ultimately your candidate drug. So this kind of surveillance is very useful pretty much at all stages of discovery, development, and also clinical development. Finally, at some point or other, you will have to do your own surveillance yourself. This is a mandatory surveillance. It's normally performed sort of late preclinical, early clinical development to really collect isolates from regions in the world where you're performing your clinical studies to really support and extrapolate data from your clinical studies. So clinical studies don't have a phenomenally large number of patients, but in a surveillance study, you can collect many thousands, tens of thousands of isolates that you can really get to grips with what is the activity of your new agent and comparators like. So that's very important. And also during regulatory approval and a few years beyond that, it is also mandatory to perform surveillance studies to track any early development of resistance. And quite often these surveillance studies continue beyond the mandatory 3 or 5 years of surveillance, which again is a useful resource if some of these data are made available in the public domain online. So to summarize, resistance surveillance is very important for hospitals themselves, for the local epidemiology, to support empirical therapy decisions and infection control, etc. Global epidemiological studies like World Health Organization, the EU, are also important for government policies, but also highlights unmet medical needs. It really gives a focus for companies developing new antimicrobial agents or wishing to, an idea of what particular drug-bug combinations might be worth pursuing. And then finally, global surveillance data. This also highlights unmet medical needs, but also supports earlier stages of development where you might be selecting a lead compound. And at later development or clinical development, it validates the data that's come from clinical studies. One other thing not to forget is that MICs are vitally important for PK/PD targets. So you need isolates with known MICs to be able to assess these factors. And also, ultimately, MICs themselves from surveillance studies are vital for breakpoint setting. Thank you.
Thank you. I'm really delighted to be here. My name is Rebecca Lee, and I lead the Vivli AMR initiative. Today, I'll be talking to you about the AMR Register. Vivli is the leading nonprofit organization. We run a global platform for clinical trial data sharing. We have over 7,000 clinical trials, and that is why, as a global nonprofit organization which runs the leading platform for clinical trial data sharing, Wellcome Trust selected us to manage this effort for an industry surveillance effort called the Vivli AMR Register. The AMR Register the mission of this is really to share AMR surveillance data in a single platform to bridge the silos between academic, private, and public information. The AMR Register is one of a number of currently externally funded projects under the Vivli portfolio. To access the AMR Register, you would go to amr.vivli.org. The AMR Register is an innovative, fully searchable platform for high-quality industry surveillance data, and I'll introduce you to this today. The purpose of the AMR Register is really to detect trends in multi-drug surveillance, multi-drug resistance over time, inform national and international policy and antibiotic stewardship, allow modeling of future resistance trends. The reason that pharma has committed to sharing their data in the AMR Register is, first of all, members of the AMR Industry Alliance have committed to sharing their surveillance data because of the Davos Declaration and the industry roadmap in combating AMR. There's really an opportunity to bring in more pharma and diagnostic companies into the AMR Register, and that is our hope. Secondly, access to medicine AMR benchmark scorecard includes antimicrobial surveillance with maximum points awarded for those companies that decide to share their raw data in an open platform such as the AMR Register. Thirdly, in 2021, the G7 communique also includes AMR as one of their important action points. Our key objectives for the AMR Register really mirror these important points. First of all, the AMR Register enables industry to fulfill their commitments to surveillance data sharing in the Davos Declaration. Secondly, it provides researchers with a single platform where they're able to openly access high-quality industry data. And thirdly, access to surveillance data really help researchers, governments, and healthcare professionals understand the spread and impact of drug-resistant infections on human health. We have an SAB that has been helping advise us throughout the development of the platform from a scientific perspective, advise us on the value proposition, and also advise us on the outreach to researchers. Mark Mendelson has been the chair of the SAB since our inception. We're also pleased to have a number of outstanding researchers, as well as Janet Medega from the Wellcome on our SAB. Let's talk a bit about the actual data in the register. First of all, I know Ian has talked quite extensively about the surveillance programs themselves, which are conducted by industry that are mandated to comply with regulatory agency programs from, for example, FDA and EMA requirements for commercial approval for new antimicrobials. This post-approval surveillance must be conducted over a period of years, as this is an important tool for industry to monitor the development of resistance to new agents as they're approved. Industry data sets include MIC data and are generated by central labs using reference methods. This is the only data that we include in the AMR Register. In addition to new antimicrobials, we also include numerous comparators that are tested alongside these new antimicrobials. And this gives us a more complete picture of the state of AMR. So these include MIC data, drug-bug pairs for the sponsor's new drug, and then we also have a number of other demographic data that's also included for researchers, such as gender, age, and the years that the data has been collected. So when a researcher accesses this data, we provide the raw data available in an Excel format. We're very grateful for our steering committee that are the data contributors that have provided their surveillance data for the register. Shown on this slide are the data sets currently available in the register. This includes the data from over 1 million isolates collected over the last 20 years from approximately 90 countries. This includes many of which include data from both genotype data and pediatric data from over approximately 20 LMICs, many of whom are from sub-Saharan Africa. I'll show you a bit about what the register looks like in just a few slides. So when you navigate to the AMR Register Surveillance Program on the website, this is what you'll see. And there'll be a few program pull-down menus showing who the members are, what their surveillance programs look like, and then how you can request the data. One of the more important pages is the actual surveillance programs and the details about them. So this is the surveillance program page. And you can see detailing, for example, what the programs themselves. Next, I'll show you the Pfizer Atlas page. So Pfizer is one of the larger data sets that we have. And as you can see here, the Pfizer Atlas page shows the countries the data is collected from, the isolate collection method, the methodology, the number of isolates, as well as whether it contains pediatric and genotype data, which it does, the date of the last update, and also any other publications that is linked to this Pfizer Atlas. So how does this actually work? So if a researcher conducts a search for data sets, for example, if a researcher is interested in data from Malawi, for example, they would do a search for that. Now, they would not simply get the data from Malawi, but which data contains data from Malawi, and they would come back with the entire data set. So for example, data set alpha and data set omega that contains data from Malawi. They could then download that data, access that data, and analyze and publish on it. Again, that is the link, search amr@vivli.org. Here's the front page of the search. You can see on what they can search for, the data contributor, the country, regions, years of collection, and whether you're interested in genotype or pediatric data as well. The organisms, for example. Well, a few things that we look for is a summary of background and summary of the proposal, the purpose of the analysis. Those are some things that we're looking for to put into the request for data. In terms of just overviewing what's happened in the last year, we've had 94 requests submitted, and many of them include requests for more than one sponsor's data, what we call multi-sponsor, and about 49 of them have included requests for a single sponsor's data. Just to dig into that a bit more, most of them are from academic investigators, some from commercial or government, NGOs, not-for-profits, and researchers from 29 countries have requested data, including 15 LMICs that are primarily interested in data from their own countries. These are the types of requests received: drug-resistant trends and profiling, to strengthen stewardship, inform local and public health policies, inform prescribing practices, for example. And these are just some examples of why researchers are interested in this surveillance data from industry. So you can go on at any time and look at the requests that have been fulfilled. Next, I'd like to hand it over to Dr. Bradford, who will talk a bit about our AMR data challenges that are now currently underway. Thank you.
Thank you, Rebecca. I would like to talk to you about the data use challenges that have been sponsored by Vivli. First, I'll start with the data challenge that we ran last year, and then talk about the new challenge for 2024. So in 2023, Vivli launched the AMR data challenge that was meant to engage new researchers in the scientific community, generate new insights based upon AMR surveillance data, and also to promote and encourage more industry partners to share their surveillance data. And these are the participating data contributors. So these data sets were made available for the purpose of the data challenge. And we had we were quite delighted with the response that we received. There were 56 entries for the data challenge from 28 different countries. From those, 11 finalists were chosen and 5 teams were awarded prizes. This is the grand prize. These are the grand prize winners, a team from Kenya, who were really an overwhelming favorites as far as the winners, because they had not only something novel and interesting that they created with the data sets, but also had a real purpose to what they were doing. They created an AI platform where a web app that will help predict local susceptibilities from using the data that they have. So their real goal was, there is a lack of information about susceptibility trends in LMICs, and of course their particular interest is in sub-Saharan Africa. So they used this AI tool where they could predict, where somebody could go in, put in an organism, and it could predict whether it would be susceptible or not to a drug of choice. This is the interface for their web app that's freely available. You can go in there and you can put in your location and what the organism is, and it will tell you whether or not it is that organism is likely to be susceptible to a particular drug. And the real purpose of their tool is to really help prescribers and antimicrobial stewardship committees where they don't actually have a lot of data from their local country. And one of the things that's really was most heartwarming from their prize winning is that they have reinvested the prize money into futuring their research. So they have used the prize money to purchase new computing systems to continue on with the AI tools. They've really taken to heart mentorship and have purchased a laptop for their mentee. They're continuing to do product development, incorporating new features, and that they have announced new partnerships and hope that this can continue in the future. So really rewarding experience for them. We had four sub-winners that were in 2 categories. The Impact Award winners were teams that really came up with an idea that would be of use to an end user, that there was really some concrete purpose to their research, that they were producing something that a prescriber could use or a stewardship committee could use. The top left team did a project that used the AMR industry data from Vivli along with some other publicly available data such as what's on glass, and came up with a way to help to synergize that information. The bottom left team was really interested in pediatrics, and so they developed some models about how you could use this data to look at whether or not isolates should be susceptible and what the trends are in from pediatric patients. The top right challenge looked at the difference between CLSI and UCAS spray points. and they examined isolates that were and MICs where there would be a change in susceptibility category based upon some of the differences that are in those two breakpoint setting agencies, and where some of those isolates, it might be troublesome for some of those differences. And then the bottom right team looked at climate. They were interested in climate change, and so they looked at where the isolate was originated, what country they came from, and what the mean temperature of that country is, and looked at the differences in resistance between temperate and tropical climates, etc. So very interesting projects. These, all of the finalists were invited to submit a paper that is in the process of being published on Wellcome Open Trust Research. So you can go on the Wellcome website and see the papers that were the output of those projects. And here's a list. There's a listing there that you go in and look. And now I'd like to talk about the current challenge that we're doing it. It was such a success last year. We're doing the data challenge again in 2024. So we had the same three goals for the data challenge, which was launched on June 17th of this year. And here's the panel list of judges. It's a really diverse panel from industry and public health and academic medicine. The judging criteria are as follows: that the entries will all be scored for methodology and study design. Is it, were they using proper techniques for how they were studying the data? But then they'll be judged on two major criteria. First, innovation. How creative is this idea? Is it something really new? Is it a new idea of how to use the data? And then secondly, an impact. Is this impact, is it going to have some usefulness to prescribers or stewardship committees, etc.? And then the prizes are as follows. The grand prize award winner will receive $10,000 and $5,000 to present at either the ECMED meeting or ASM Microbe in 2025. There will be 4 sub-awards of $5,000 plus a $2,500 travel grant, 2 awards each for innovation and impact. And we're adding a new award this year, which is a Student Innovation Award, which is $5,000 to travel to present their abstract at one of the meetings. And this team must be made up primarily of students with 1 PI mentor on the team. So we're really excited to see what we get from the student category. And then finally, all of the finalists will have to post their solutions in an open source forum. So here's the timeline of what will happen with the data challenge. We had the data challenge was announced in June. We've just announced that we are going to extend the deadline for submission of interest. So you all have 3 more weeks to submit an expression of interest. And this is a 300-word, just an outline basically of what your project will entail and what you hope to achieve out of doing the exercise. Once the expression of interest is approved, then you will receive, you'll be able to download the data, and then you have 30 days to make analyze the data sets and post your final solution. So after 30 days, you'll submit your final solution. This is not a, it's not a tremendous document that we're expecting. It's a, 5-page summary, usually with some figures and tables of what the output is from the data analysis. And then sometime in October, we'll have live judging with the panel, with the judges, and this is done over some video conference platform. And so we'll have a face-to-face time with teams to have them present their findings. If they have built some kind of a web tool or an app, that they're asked to do a live demo during that time, and then they'll be judged. And then in early November, the prizes will be chosen. And then in 2025, they will present them at the various conferences and hopefully publish again in Welcome Open Research. So this is where you can go in and find out about the data challenge. So if you go on the AMR Register website, in the upper right, there's a pull-down menu for the data challenge, and this has all the information that you need. And then just closing thoughts about the AMR Register. It's an important free resource of industry surveillance data, and that is continually updated. We do welcome interested researchers to join the data challenge that's currently open, and resources are available online if you have questions, and our team is always available to answer further questions. Thank you.